Okay, so can you see my screen, people?
Yes, sir.
Okay, so uh, this is a topic that many of you, particularly the ones who had biology in class 12th, have probably already seen. This is very simple, and yet there are lots and lots of people who have major conceptual issues with this, deceptively simple law. So even if you know what the final form of the equation will be, it's still worth seeing why you end up where you end up and why this is supposed to be a big deal. For those people who are not aware of it, I'll be starting from scratch. So if you remember, I said that there was one major issue with Darwin's work, and that issue was that he didn't have a good, credible mechanism for inheritance. And that issue gets resolved when Mendel comes into the picture. And Mendel essentially gave a particulate theory of inheritance, which basically meant it was not blending. That way, the variation would still be maintained. However, if you remember Mendel's work, Mendel was studying families, right? He took a tall plant and a short plant, and then he crossed them, and so on and so forth for about seven other traits. So whatever ratios he was getting, 1:3, 1:, uh, you know, 1:2:1, all those ratios are in that context. However, as we saw in the last class, when you talk about evolution, evolution is at the level of the population. So if you are thinking of mathematizing the whole thing, formalizing all these concepts that we have been describing, then you need a scenario, or you need a way by which the laws of Mendel at the family level, they can be taken to the level of the population. Okay. But before we do that, we need to quickly revisit what Mendel's laws were. So if you remember, there were two major laws. One was the law of segregation. I mean, the law of dominance is also there, but we don't need it over here. One is the law of segregation, which says that in diploid organisms, when the gamete is formed, then each gamete is going to receive only one copy of the allele. Normally, otherwise, they will have two copies. And the second was the law of independent assortment, which said that during gametogenesis, the alleles of different genes, they assort independent of each other, which means they behave as if you picked one of them, one of the alleles at a locus and another allele at a different locus, independent of each other. We now know that Mendel got incredibly lucky because the traits that he was studying, those traits were on either on different chromosomes, or when they were on the same chromosome, they were very, very far apart, because of which linkage did not complicate his his calculations. Otherwise, when the loci lie close to each other on the same chromosome, then there is a probability with which they get separated from each other. The closer they are, the higher the probability that they will be inherited together. So this independent assortment breaks down when you have linkage. So what is the problem? Okay, the problem is that, as I said, you need to go from transmission of genes in a family to transmission of genes at the population level. So what, and that is the subject of population genetics. So what population genetics is trying to explain is microevolutionary changes, changes in terms of gene frequency. So you have a population at time t, and this population at time t, its gene frequency is going to change, and then you are going to see the population at time t plus 1. And you need to figure out how these changes happen. Now, the population is composed of individuals, right? So at the level of the individual, underneath the hood, so to speak, what is happening is this. You have the male individual. In the male individual, during the process of meiosis, two things are happening. A, because of recombination, the genes are coming in different combinations with each other, the various loci. And then because of reduction division, only half of those alleles, half of those genes are being passed to a gamete. So each individual, a male individual or a female individual's contribution to the next generation is not the full genome, half of the genome, and that half is also, let's say a scrambled up half. It's a messed up is the wrong term. It's a tossed up kind of a half that goes to the next generation. And then these two are going to fuse with each other to form the zygote, and then the zygote will undergo the process of development to form the juvenile, and then the juvenile will mature to form the individual. And then you have to track everything, all whatever is happening at the level of each individual. Then you sum all of them up and you get to your whatever is the gene frequency in the next generation, right? So what appears to be a simple one arrow over here, underneath is this much amount of complication. And also remember, when you have a male individual and a female individual, not everyone is mating with everyone, right? So which male will end up mating with which female is going to affect all these complications in terms of which gene is going where. So obviously, this is not something that you can do at an individual by individual level the way Mendel was doing, right? So it is going to be something that is going to be more of a probabilistic, a statistical kind of a thing. And the subject which does that takes Mendel's laws and extends them to the level of the population. That subject is what is called population genetics. So in population genetics, we explicitly look at the allele frequencies, how they are changing across time. There is one more subject called quantitative genetics, which takes a different approach altogether. It takes the approach of taking the statistical distribution of the traits and tracing how that distribution of traits is changing over time. So here you look at alleles, there you look at traits. The two subjects are actually, they talk to each other. They are very nicely connected, by the courtesy, the work of Ronald Fisher. But we won't go to QG. We'll just take into PG for this particular subject, this particular course. So here, how exactly does population genetics do it? Now, in order to do that, population genetics makes a certain, it conceptualizes the whole thing in a particular way. And this conceptualization, understanding this conceptualization is the key over here. So all of you are able to see the screen, right? This white screen over here.
Yes, sir.
Yes, sir.
Okay. So if you think about it, a given individual is going to have hundreds or thousands of genes depending on which organism you are looking at, right? Oh. So population genetics, when it considers itself with one gene and alleles at that gene, then it basically treats each individual organism just as a pair of those two genes, those two alleles. So for example, essentially, this is what population genetics will conceptualize an individual as. So this is, let's assume for a moment that the two genes that we have in hand, one of them is black and the other one is white. Okay. So let's assume we have a simple case where we have a one locus, two allele scenario. Once you see the conceptualization, you will understand that this, you can very easily go more than this. This is just for the sake of illustration. So suppose this is my population, and inside this population I have multiple individuals. So each individual is basically just regarded as a pair of genes. That's it. Okay. the size difference has no meaning. It's just that I'm trying to fit in more of those, and that's it. So if that be the case, if I have one locus and two allele, and one of the alleles is white and the other allele is black, then this is what my population is going to look like, correct? Some stuff like this. Now, the process of meiosis is conceptualized as the coming apart of these things. So this is the diploid individual, right? So this is the population at the adult level. The population at the gamete level, then becomes simply like this: a bunch of white gametes and a bunch of black gametes, right? So this is the so-called gamete pool. Right? Now, let's assume for a moment that these are all the male individuals. Now you are also going to have all the female individuals who are also producing gametes of the same, white and black type, correct? So actually you have not one, but in a sexually reproducing organism, you will have two gamete pools, a male gamete pool and a female gamete pool. Now remember, there is mating happening in this particular, in a sexually reproducing organism. So somewhere these organisms are going to find each other, and based on whatever sexual selection or whatever is courtship ritual, etc., etc., these guys are going to mate with each other. So the process of mating then gets abstracted out by pulling one gamete from here and one gamete from here, let's say this is a black gamete, and then joining the two. Okay. So this picking up, you can either do it randomly, in which case this is just like closing your eyes and putting your hand inside a bag full of let's say different colored balls, or you can have some rules of picking by which, let's say, from the females, let's say the chance of picking up a black ball is much higher, or a black gamete is much higher, whereas suppose from here the chances of picking up a white gamete is much higher, or vice versa, whatever, any kind of crazy rules that you can pick. But essentially the process of mating, fertilization, and formation of zygote, that entire thing essentially is drawing some balls from here, from the male pool, some from the female pool, joining them together and reforming the zygote population. Okay.
Sir,
Bolo.
Sir, can we also take into consideration the spatial probabilities? So if
anything into consideration, anything that will dictate in what way this pull is happening is fair game. But the point is that conceptually speaking, the gametes coming together, fusing, fertilizing, and forming a zygote boils down to pulling from this one, pulling from the male gamete pool, and pulling from the female gamete. Okay. Now think about this. If we are thinking of it in this particular way, there is a massive amount of biology that is just being swept under the carpet, right? There is no behavior, there is no morphology, there is no development, there is pretty much nothing. All that we are saying is you have a bunch of coupled balls in parental generation, you have a bunch of dissociated balls in the next generation, in the gamete pool, and then to form the next generation, just draw from the male gamete pool, the male bag, just draw from the female gamete pool, the female bag, and then put them together to get the zygotes in the next generation. Okay. So essentially, this is tracking just what's happening to the alleles, not considering what is happening to the rest of the body, rest of the genome, rest of the biology, the environment, everything is just swept out, right? So now I'll go back to the presentation. Is this conceptualization making sense? Why this conceptualization, or rather how this conceptualization is being done?
Yes, sir.
Everybody?
Yes, sir, it makes sense.
Okay. So we'll now go back to this thing. So, so this is what I was saying, that at an intuitive level, the individuals are regarded as gene pairs. The gametes then simply become the single genes, what I call was the dissociated balls. Mating and fertilization is coming together of those gametes, and the next generation is formed by sampling from this pool of gametes, right? So this step. So this thing, this, what I just now drew on that canvas, those things look like these beans that we have, no? What bean seeds? What are they called? Rajma, and what is that other thing? I've forgotten those. अरे यार, white आता है और ये आता है, white and red they call it. Anyway, whatever. You guys get rajma, right? So some kind of beans. Huh?
Kidney beans.
Kidney beans is one, but kidney beans is rajma. But there is one more thing which is smaller in size.
Lobia.
Lobia. Right. Lobia. Thank you. Lobia was the name I was searching for. So all those are different kinds of beans. So essentially, the whole thing looks like sampling from bags full of beans, and that is why this kind of genetics is derisively called beanbag genetics. Okay, it's like a bunch of accountants trying to keep track of who gets what colored and in what frequency. So this, so the entire process of transmission of genes over a large number of mating pairs, it gets abstracted into sampling from a bag full of gametes. Okay, so this conceptualization is actually extremely powerful, although deceptively simple. But this conceptualization allows you to basically figure out much of the mathematics of microevolutionary processes. Okay, as I said, this is the point at which population genetics begins. So let's make this a little bit more formal. All right. So let's put some numbers over here. So let's assume that we have a one locus two allele scenario. Okay, so we have a one locus, and that at that one locus we are considering only two alleles. Okay. Now let's assume that the number of individuals in the population is n. Okay, so let's assume that our two alleles are A1 and A2. Then Mendel's laws tell us that we are going to have three genotypes: A1A1, A1A2, and A2A2. Okay. So let's further assume that the frequency of A1 is equal to P. So if the frequency of A1 is P, then, and okay, let's also assume that the frequency of A2 is equal to Q, right? Now, since there are only two kinds of alleles in the population, therefore P plus Q, small p plus small q, is always going to be equal to 1. Does that make sense, everybody?
Yes, sir.
So P plus Q is equal to 1, which implies that Q is equal to 1 minus P, or P is equal to 1 minus Q trivially. Okay. Now, let's further assume that the frequencies of these A1, 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 A2, A2, A2 are given as P, Q, and R. Capital P, capital Q, and capital R. Then it can be trivially shown that small p is equal to p plus half capital Q, and small q is equal to half Q plus R. Do you guys see why that is so trivially shown? Is this intuitive to everyone, or shall I spell it out? Okay. So think about it. We're talking about 1 locus 2 alleles, so we have a diploid individual case here, right? Now I said that there are n number of individuals in the population. So if there are n number of individuals in the population,
Sir, it makes sense.
let me spell it out. I mean, if you think about it, it will make sense. Let me spell it out anyway. So if there are, is it making sense to everybody, or do people want me to do the spelling it out?
No, it's making sense.
Everyone?
Okay.
I'm
I
not
think-
getting it.
Sir, why is it half Q?
Okay. Okay, then you need to see this. Why is it half Q? So if there are n number of individuals, then the total number of alleles in that population is 2n, right? Some of which are type A1 and some of which are type A2, but their total number is going to be 2n, correct?
Yes, sir.
Now, let's think about the guys who have the this A1 allele. So the A1 allele is present in A1A1 individuals, and they are present in A1A2 individuals, correct? So how many A1 alleles do I have in the population? Okay, I have to put in one more thing. So let's say that the number of individuals of type A1, let's say this is X, let's say this is Y, and let's say this is Z. Okay, so there are X number of A1A1, Y number of A1A2, and Z number of A2A, right? So if that be the case, then the total number of A1 alleles equal to 2x plus y. Does that make sense?
Yes,
Each
sir.
x individual contributes 2.
Yes,
These
sir.
guys contribute 1. So it is 2x plus y, correct? So if I want to know the frequency of the A1 allele in the population, so frequency of A1 is equal to 2x plus y divided by total number of alleles, which I said is 2n, correct? This thing is equal to x by n plus half of y by n, right?
Yes.
But x by n is what? X by n is the frequency of these A1 individuals, which is p, correct?
Okay. Yeah.
So this is p, this is again half, and y by n is q, right? And frequency of A1 in the population is small p,
Yes, sir.
correct? So that is how you get this stuff over here. Small p is equal to p plus half q by r, and just similarly in the other direction, small q is equal to half q by r. Okay, everybody, makes sense?
Yes, sir.
Cool.
Sir, should we not call it not as frequency but as ratio, or is it the same?
In this particular context, it's the same, no, because I have 2n over here, so total number of alleles is 2n, and I have what was it, 2x plus y over here.
Yes.
So
Okay.
this is the frequency, right? How many are out of the total, how many are of type A1? So that is the frequency of A1 by definition. It's not a frequency in that other sense that you are talking about.
That made me confused. Like we used to count now, so that we used to say as frequency. So
Right,
it's
right.
ratio.
yeah, so but in the population genetics literature, this is called frequency. Okay, gene frequency or allele frequency.
Okay, sir. Thank you.
So you need to keep that thing in mind. That is what I have said here, that small p plus small q is equal to 1 trivially. Since there are only 3 genotypes, therefore the ratios, the frequencies are going to sum to 1. So that's p plus q plus r is equal to 1. And there is a very simple relationship between small p and capital P, capital Q, capital R. And this is that relationship. P is equal to p plus half q, q is equal to r plus half q. Now note something. When we did this, we did not make any assumptions whatsoever about the population structure, about the population's mating or anything. Okay, as long as the population is diploid and as long as there are those many number of alleles at the one locus, this particular thing is going to hold. This is a very, very important relationship, and lots of people actually mess up, and I'll show you how the mess up happens in not understanding this, how this thing works out. So just to give you guys an example, suppose there is a population in which you have 83 individuals who are capital M, 46 capital M N, and 11 capital N, capital N. Okay. So can you guys quickly compute the gene and the genotypic frequencies? I hope you guys have some pen and a paper in front of you. People, are you guys there? Is anyone even trying?
Yeah, sir, we're here and people are also answering in the chat box.
Hey guys, you have to realize that I can't see the chat box when the screen is full screen like it is now. So somebody has to speak out.
Sir,
Huh.
so for M, the gene frequency is around 0.75, and
Okay.
and so N will be just 1 minus M.
1 minus that, yes.
So it will be 0.25.
Yeah. So this is what it boils down to. And if you are somewhere in the ballpark range, and hopefully you have gone by this thing, then it should be okay. All right. So now we are trying to derive what is known as the Hardy-Weinberg relationship. So let there be two alleles, A1 and A2 at one locus, and let the frequency of A1 be small p. Okay, exactly what we took in the last slide. So if that be the case, by definition, what we are saying is that the frequency of A1 in parents is small p, right? So remember that diagram that I showed you guys about, going from gene frequency in generation T to gene frequency in generation T plus 1. We are going to follow that path, and at every step we are going to ask what is the gene frequency now, and in some cases what is the genotypic frequency. So right now the frequency of capital A1 in parents is small p by definition. So from this we have to go to the gametic pool, which basically means that we have to, let's assume that we have taken all the gametes of one type, say the male gametes. Suppose we have taken all the gametes, then what is going to be the frequency of A1 in all those gametes? Now, in order to get to that, suppose we have these conditions. Segregation is happening normally. So there is equal fertility. So all the genotypes are putting in equal number of genes in the population, in the gametic pool. There is no mutation happening, there is no migration happening. Okay. So if all these conditions are being met, then what will be the frequency of A1 in all these gametes in the gametic pool?
P.
Hmm?
Still.
Exactly. It still remains
Yeah.
P, right?
Hmm.
Because what is happening over here is that each parent is essentially contributing equal proportion to the next generation, to the gametic pool, sorry. Okay. So normal segregation means that the A1 and A2 are segregating properly. Each one of them is giving equal numbers. A1 is not changing to A2 or A2 is not changing into A1. No A1 is coming from outside, no A1 is going out, right? So, and equally for A2. So when all these things happen, sorry, when all these things happen, then the frequency of A1 in all the gametes remains at P, right? Now, if you look at this, this is telling you in what all ways this can go wrong, right? Now, from frequency of A1 in all gametes, we have to go to frequency of A1 in the gametes forming the zygotes. Why is that so? Because remember, a very large fraction of gametes produced by the males and a pretty large fraction of the gametes produced by the females, they never get fertilized. So only a small part of that is getting fertilized, right? So we now have to track the frequency of A1 in that small fraction in which where the fertilization is happening. Now, what are the ways in which this can become non-representative? What are the ways in which this frequency A1 can become non-representative of this?
Sir,
So,
if
Akash, continue, sorry.
no, go ahead, Prateek.
So if some animals go out of that place or something like that.
That we have already taken care over here, no migration, etc. Because remember, now we are in the gametic stage. The gametes cannot go anywhere.
If
Oh, I didn't see that, sorry.
what happens if a particular gene maybe compromises the motility of the sperms or something like that? Particular allele will not be selected for because the sperms are not motile.
Yes, but then that is what essentially comes in over here. So if a sperm is not motile, then the fertility is not equal. Okay, so here you are assuming that all the gametes are on equal footing already.
Okay.
If there's preferential mating,
Mating has still not happened. So we have, remember, this is a conceptualization happening, right? So we have all the gametes which have formed the gametic pool, and from that we are going to pick up a set of gametes which will now fertilize the other set of gametes. So suppose this is the male bag. So from the male bag we'll pick up something which will then pair with the female bag. That pairing level, the mating will come into picture. Okay. Right now we are just selecting all those gametes which will form the male fertilizing population.
Suppose if there is a gene which suppresses this gene in some way.
Which gene suppresses which gene?
Some gene suppresses A1.
I mean, Baba, this, see, A1 ke function, there is nothing happening in the context of the function of A1 over here. All we are talking about is the frequency. What numbers? That's all. The function enters nowhere in the picture. Remember, we're talking inheritance. No, inheritance mein function doesn't come into the picture directly.
The gene number.
Huh? Yeah, go on.
The number of genes are not distributed uniformly, maybe. Like it's not n and n plus 1 and n minus 1 in some cases.
See, in this context, as I said, we are just talking about one locus, right? So that is when you forget about everything that is happening at the other loci, right?
Mm-hmm. Yes.
So you have to think in terms of the picture that I view for you guys. See, the point is
sorry, like biological, I don't know, but there could be a sampling bias while we are moving from the second one to the third picture.
Exactly. So here you have all the gametes that have been produced by the males. Okay. Which means, given, let's say, each male produces a few million gametes each time. So this is like a huge 10 to the power 9, 10 to the power 10, some number like that. From that, you are going to the frequency of only those, or the number of only those gametes which are forming the zygotes. So you are doing a massive sampling, right? So whenever you do sampling, what do you have? A possibility of a sampling error, correct? So how do you minimize sampling error? How do you make a sample more and more representative of the population?
By
Take a
taking more
large, a
samples.
very large population.
Exactly. In
But
the
sir,
lineage,
then randomness is like built into the system, no?
yes.
If we are trying to predict something, something else can happen because.
So
Okay, I was just wondering if we are making models out of this, but we sample it some in some other way, we'll end up getting some other trait distribution, something.
see, that is always the case. Whenever you are doing any kind of sample, one run of it is always or typically going to be different from another run, right? So for example, if you toss a coin, you can either get head or you can get tail, but the probability of getting a head or a tail remains the same, right? So we will
Yes.
be talking about that probability. We will not talk about what happens in a particular instance of it. Okay.
Oh,
So frequency of A1 in all gametes going here. If we want to maintain that frequency at P, then we need to have a very, very large sample. Now, how large is large? Theoretically speaking, this has to be infinitely large, right? Number one. And number two, okay, I think this is what Kash, you were probably talking about, the fertilizing capacity of all the gametes. Okay, I think that's where your, I don't know who's talked about motility and all that.
Yeah,
All
yeah.
right.
That,
Okay.
that was one.
I was thinking about it over here. You can also think about it over here. I think this is explicitly taking care of that point. Equal fertilizing capacity of all the gametes, right? So if you have ensured that these two stuffs have been met, then by what we discussed, frequency of A1 in gametes forming zygote still remains at P, correct? Now, these gametes are going to form the zygotes. So from this, now from frequency of allele, you have to go to the frequency of genotypes, right? Because zygotes, remember, are diploid. So you can't think of allele frequency in zygotes, you have to think in terms of genotype frequency. Now, this is where the mating part comes in. Now, suppose you ensure that mating is random and you ensure that the frequency amongst the males and the females remains the same. Okay. If that be the case, so think about it this way. So you have the male bag and you have the female bag, correct? In this, you have, let's say, the frequency of allele A is P. Let's say in this is the allele is also P. So now you have blindly put your hand inside and you have brought out an allele. What is the chance that that allele is a A1? P, correct?
Yes, sir.
yes.
What is the chance that now you have brought out a female gamete and that female gamete is A1? What is the chance? That chance is again P, correct? So the probability of forming a A1 genotype in the next generation is P into P equal to P square. Agreed?
Yes, sir.
So this is something that should be very familiar to you guys. Remember what this is called? So the Punnett square, yes. Okay, so let's assume that these are the males. So the probability of drawing a A1 is P. The probability of drawing a A2 is Q, where Q is 1 minus P. Here it's P, here it's Q. So then the probability of making a A1 is P square. The probability of making a A1 A2 is PQ. The probability of making a A2A1, which again, if you don't consider ordered genotypes, then it is again A1, A2, is again PQ, and the probability of making a A2A2 is Q square. So two PQ, because I'm adding these two, is this, right? So remember, this thing happens if and only if you are drawing them independent of each other, which means whatever you are drawing from the male gamete pool has no effect on whatever is coming out of this gamete pool. Only then you can do this multiplication, right? If that thing is not there, the independence is not there, then you can't do this multiplication. That's one. So the independence is what the condition of random mating. And remember that when you are pulling a gamete, then the probability of pulling that particular kind of gamete is equal to its frequency in the population, right? That is the reason for which you are able to put PQ here, you are able to put P and Q here. If the probability of pulling that gamete is not the same as its frequency in the population, then what goes over here is not the frequency, what goes over here is the probability. Right? I mean, basically the entire calculation that gets messed up quite a lot, right? So you cannot put the frequency over here anymore. This is a very big thing, and this is the one that was saying that each allele has got a probability of getting pulled up, which is simply determined by its frequency in the population. Now let's go back to this. Okay. If all these things happen, then disregard this plus sign. It's actually not a plus sign. Okay. So if this thing happens, then the genotypic frequency among the zygotes, then the genotypic frequency among the zygotes becomes P square, 2PQ and Q square. Now, all the zygotes are not going to develop into progenies, only a fraction of them will become progenies. So under what condition will this frequency be maintained?
Large sample size again.
If all survive.
If all survive, or if all of them die at equal rates, right?
Yeah.
So if there is 20% mortality, everybody is suffering from the same 20% mortality. So that is what is called viability selection. So note over here that selection is coming in two different ways over here. One, it is coming when we are talking about it over here, going from zygote to progeny. It also comes here. Okay, going from okay, sorry, it only comes there. Sorry. So it comes over here at this stage, and it also comes over here when we go from A1 in parents to A1 in among all the gametes. But the way it is going is slightly different. Here it is going through how many gametes you are producing. Here it is going through whether you are able to survive till the point of reproduction in the next generation. Okay? So the way these two kinds of selection are treated is different. This is what is called as fertility selection, and this is what is called as viability selection. This is easier to treat. This is slightly more difficult to treat. So in our treatment, not today, in let's say tomorrow or day after, we are going to look at this kind of selection. Okay. So if that happens, then the genotypic frequency among the progeny remains P squared to PQ squared. Now, what is going to be the frequency of A1 in progeny? Remember, we have started with A1 in parents, so we have to go to A1 in progeny. Now, the frequency of A1 in progeny is going to be, remember, small p is P plus half Q, correct? So small p in the next generation is going to be P dash is equal to P dash plus half Q, equal to, what is my P dash? This is P square, small p square, plus half Q means PQ, Correct? This thing, half of that
Yes.
equal to, correct? Equal to P plus Q is always 1 trivially. Correct?
Yes, sir.
You guys see how this is coming? So,
Sir, did you
Abdullah,
basically just say that? Did you essentially say that if nothing interesting happens, then nothing changes?
yes.
Okay.
I said if nothing interesting happens, nothing changes. But in the process of saying that, I actually said, I gave you guys a full laundry list in everything interesting that can possibly happen over here. Do you guys see that part?
Yeah. Yeah. Okay.
Okay. So, statement of the law. In a large random mating population with no selection, mutation, or migration, the gene frequencies and the genotypic frequencies are constant from generation to generation, and there is a simple relationship between the gene frequencies and the genotypic frequencies. Now note, lots of people, when they are asked what the Hardy-Weinberg law is, they simply say P plus Q equal to 1 and P square plus Q, 2PQ plus Q square is equal to 1. The problem is, in a 1 locus 2 allele scenario, P plus Q will be always equal to 1. P square plus 2PQ plus Q square will be always equal to 1. There is nothing else that can happen to those two things given their definition. Okay, so you don't really need the name of two big scientists to be associated with something that trivial.
So
just one second. The real statement of the Hardy-Weinberg is this one. Bolo.
So, I mean, along the same lines, what Akash had asked. So, if you have all of these assumptions, then whatever the thing is, P square, 2P, Q square, that is a simple combinatorics problem, right? Then
Absolutely.
why is it a big thing, such a big
Why
deal?
is it a big deal? Fantastic problem. And that is what I need to explain next. I don't have time today, so I'll stop the class right now. And tomorrow, I will explain why this is such a big deal, why this is supposed to be like the cornerstone on which the entire subject of population genetics is built. Okay, if you guys hang around for some time today, I'll give you guys a quick answer, but formally the answer will have to wait till tomorrow because I have run out of time today. Okay, it's a good question. Okay, so I'm going to stop the recording today. Adrian Pinto, are you there? Adrian Pinto? Adrian, this is going into your hard drive. So please,